Yes.
Okay, I must apologize in advance. I'm a little under the weather, so my voice is going to flicker a little bit. I hope I'll make it through the end of the talk. So thank you for the invitation. It's a pleasure for me to be here today and giving you an application talk on number and brightness and show you how that technique is changing glucocorticoid receptor biology. So glucocorticoids are extremely important from a pharmacological perspective. You might have heard the name dexamethasone last year. It made a lot of headlines because it was one of the first successful drugs to treat severe cases of COVID-19. And that is because glucocorticoids are extremely good at anti-inflammatory and immunosuppressive activity. So I can use some right now, but unfortunately glucocorticoid treatments, especially the chronic one, has severe adverse reactions. So one of the, like the ones you can see on this table, excuse me. So one of the objectives from a pharmacological perspective is try to find these dissociated ligands. That will mean glucocorticoids that will keep the desired therapeutic effects, but will reduce significantly the adverse reactions. So taking that into account, a model has been built on glucocorticoid receptor action called the dissociated model of GR action. So glucocorticoids bind to a protein called the glucocorticoid receptor, which is a ligand-regulated transcription factor. And the dissociated model states that GR, which is in this cartoon, it's a protein that has 3 structural and functional domains. It has an N-terminal domain, which is intrinsically disordered, a DNA binding domain, which is the part that binds DNA, and a ligand binding domain, which is the part that binds ligand and interacts with several cofactors. So according to this model, GR lives in 2 oligomerization states. It's either a monomer or a dimer. And the dimer is the one that is involved in binding directly to response elements across the genome and then regulate gene expression that way. That was historically named the transactivation activity of the receptor. And that is believed, according to this model, to be involved in most of the side effects of glucocorticoids, okay? Well, the monomeric receptor, what it does is that it binds indirectly through DNA, to DNA, by protein-protein interactions with other transcription factors, like NF-kappa B or AP1, which are major players in the inflammatory response. So in this way, monomeric GR acts as a co-repressor of this transcription factor, historically named the trans-repression activity of the receptor, and it's believed, according to this model, to be involved in most of the inflammatory reactions. So in a nutshell, the dissociated model predicts that it is the monomeric GR who triggers the anti-inflammatory actions of DNA in a DNA-dependent manner. So if you want to find better glucocorticoids, you should try to find ligands that will shift the equilibrium towards a monomeric receptor, okay? So, and that was consolidated by studying a single point mutation in the receptor called the GRdim mutant. So the GRdim mutant is a point mutation in the DNA binding domain. So these are the 2 zinc fingers of the protein, and the mutation is right here. And that protein, that mutation, is supposed to impair dimerization and DNA binding, okay? So if this receptor cannot dimerize nor bind DNA, then it cannot do anything of the transactivation, right? And it can only do transrepression. So the big surprise was that it was known a few years earlier that a GR knockout was lethal, but the GRdim survived. And that's because this protein could not bind DNA. This gives basically the title of the paper, that the direct DNA binding activity of GR was not essential for survival. And then there are dozens of papers just using GRdim as a tool to know if your pathway is basically, you know, monomeric DNA independent or if it's dimeric DNA dependent, whether it happens on the DIM or not. And then later on, it was just straightforward, suggested that this would be a way to find and treat all inflammatory diseases by trying to select the activity of DNA-independent activity of GR, okay? However, the original paper that described this mutation, you look at every single figure of the paper and you will not find any direct nor indirect evidence of the oligomerization status of the GRdim protein, okay? So that's when we start trying to contribute this. So we use number and brightness to measure the oligomerization state of GR. So this, you heard a beautiful explanation early on, but I just gonna give you a brief summary. So this technique uses a fluorescence intensity fluctuation to estimate the oligomeric state of proteins in live cells. So if you just measure one single point in the cell and you have either monomers, dimers, or trimers, and these molecules are free to move in and inside and outside this excitation volume, you will measure this kind of intensity patterns where the higher the oligomeric states, the higher the amplitude of the fluctuation that these molecules produce. And you can distinguish these situations by measuring the variance of these intensity fluctuations. So a monomer will have a certain amount of counts per molecule, and that will depend on acquisition parameters like laser power as it was described early on. But if you, and in relative terms, so a dimer which has 2 fluorophores will have twice the amount of brightness at the case of the monomer, and the trimer will have 3 times the brightness on average than the monomer. So the implementation that we use for GR was to take images in a confocal microscope in the same way that you will do a RIGS experiment. And then you will select the pixels that correspond to the region of interest. In this case, is whatever is going to happen inside the nucleus of the cell. And then for each pixel of inside the nucleus, you can estimate the variance and intensities. You can calculate the brightness. And then we will get the distribution in the entire nucleus. So we will retrieve some sort of an average nuclear brightness that will give you an overall idea of the oligomerization state in the nucleus. One important, and so therefore we can plot this average nuclear brightness for many cells. So in relative terms, if we know a monomeric control, it's going to be relative to 1. So we know the dimer will have twice the brightness of the monomers and so forth. So one of the caveats of this technique is that it cannot discriminate mixtures of oligomerization states. So what do I mean by that? So if we do have a mixture, let's say monomers and dimers within the same region of the cell, then you're going to receive a weighted average brightness. So you will have an intermediate value between 1 and 2, depending on the relative proportion of each of these species, okay? So let's take a look at GR data, okay? So here is, again, the nuclear brightness plotted relative to 1 being the monomer. That's what happens with the receptor in the absence of hormone, which we compare it with GFP, which is a monomeric control, and we basically have the same result. So once we activate with hormone, then the wild type receptor changes the brightness from 1 to 2. So that not only means that GR is dimeric inside the nucleus of living cells, it also means that most of the molecules must be dimeric. Otherwise, you will, if you have, for example, a significant population of monomers, you will receive an intermediate value between 1 and 2. And we can actually test that by doing a control experiment. So now we can put in the same cell 2 types of GR, one tagged with GFP and one tagged with M-cherry. So you're going to see basically these complexes form. We're going to see GFP homodimers, M-cherry homodimers, and these GFP M-cherry heterodimers. So if we only look at the green channel now, these guys, we're going to see them as dimers. These guys were not even going to see them. And these guys, even though they are dimers, we're going to perceive them as monomers because they only have 1 GFP molecule per complex, okay? So when we do the experiments, we obtain more or less what we expected, which is an intermediate value between 1 and 2. And this exact value will depend on the relative ratio inside each cell for each of these species, okay? So one of the hallmarks of the dissociating model is that GR interacts with NF-kappa B as a monomer. So if that's true, and if in the same cell we have GR and we also have P65, which is one of the subunits of this protein, of this transcription factor, and we activate both, then you will expect to see an increase in monomeric population because this is supposed to be happening. So we should expect more this result rather than this result. That's not what happens. We can still see GR mostly dimeric, So the most likely scenario is that if this is going on, it's actually GR doesn't change oligomeric state once it's interacting with this protein. But that was established based on the GRdim mutant, which was supposed to be monomeric. So we did the test and we found that GRdim remains dimeric, mostly in the nucleus. So we thought, well, since the mutation is on the DNA binding domain, maybe there is another region of the protein that must be holding the dimer together. So long story short, we found this mutation was originally described in the crystal structure description of the ligand binding domain. So we did that mutation and we already observed an equilibrium between monomers and dimers now. And when we combine both mutations, so the GRdim plus this LBD mutation, we obtain, now we're trying to affect both dimerization surfaces. I'm sorry, that was for the other case. And that's what we see now when we affect both dimerization cases is that we get a mostly monomeric GRmon with a brightness of around 1.2. So we coined this protein the GRmon mutant. So the idea that monomeric GR is the one causing transrepression is put into question. So we may have a model where either the monomer or the dimer could be doing this process, but since GR is mostly dimeric to begin with, it's more parsimonious to think that it's the dimer who's doing everything here. And even though there's been a lot of papers showing that transrepression, as I define it, it's responsible for metabolic side effects, while transactivation is needed for anti-inflammatory actions. Now there's even papers that are claiming that this process is not even happening. So I'm going to finish with just also questioning the idea of what is the oligomerization state of GR when it is bound to DNA. And that's not a trivial question to answer, especially in live cells. So what we did is we use this very interesting model that we have. So these are called 3617 cells, and they are mammary adenocarcinoma mammary cells. What you can see here is that this is GFP-GR in the absence of ligand. So you can see that it's mostly cytoplasmic. Well, something in the nucleus, but you add hormone, you're going to see that it's fully translocated, almost fully translocated to the nucleus. You're going to see these big, this little foci, these little bright spots, which Valeria is going to talk next about. But I want for now to focus on this huge blob right here. Okay, I don't know if you can see it. And what's going on in that region that has a lot of GR molecules is that inside there is an insertion of a gene cassette repeated about 200 times, which has a reporter gene and binding sites for GR. So basically here you're going to see, we know there's a lot of specific binding going on because we are highly enriched in this specific binding. So since we have a technique that measures oligomerization state with high spatial resolution, we wonder what will be the oligomerization state of GR within that, what we call this MMTV array. So these are the results for what happens inside the entire nucleus in red for either the natural ligand corticosteroid or the synthetic drug dexamethasone. So we have dimerization in the nucleus on both hormones. But the question is what happened at the array, okay? And we were extremely surprised to find that the organization state was much higher. The brightness was between 3 and 4 for the natural ligand, and it went straight to 4 for the synthetic drug. So the most parsimonious explanation that we found is that GR is most likely a tetramer at the array. So we try to rule out, try to think, well, maybe GR is a dimer, and we're seeing some sort of artifact. So I'm just going to mention one that personally convinced me that we actually had something real here. So I was looking at this paper. This author were studying the DNA binding domain fragment of the protein. So just the part that binds DNA. And here you have the conformation that it has without DNA binding by NMR in solution. And in violet, you see the structure that it has when it is bound on DNA by X-ray crystallography. So you can see everything is more or less the same here, except this part which changes, okay? So especially this, which is called the dimerization loop, which is ironically where the GRD mutation is. So all I'm saying with this is that, I don't know if you can see my hands, that GR, once it binds DNA, it just changes conformation, at least the DBD, okay? So you can think DNA as another ligand for GR that induces a conformational change. So these guys were studying these 2 independent mutations that what they do is that they lock the receptor into the DNA bound conformation, even though it is not bound to DNA, okay? And that was a fantastic mutation for us to test our hypothesis that maybe DNA was triggering some sort of higher oligomerization state like the tetramer. So we tested by NMB this mutant. So at the array, numbers were kept at 4. So the question was, what's going on in the entire nucleus? Because most of what we're measuring there are diffusive molecules. And we were extremely happy to see that now this DNA mimic mutation is sufficient to trigger tetramerization everywhere in the nucleus. So with that and another experiments and some other work from others, we came up with this model where GR is basically a dimer and it binds to DNA. There's a conformational change in DNA, in the DNA binding domain, which somehow triggers also conformation in the LBD, which is the one which is responsible ultimately for the tetramerization. And we don't know if the tetramer is a head-to-head configuration or either the head-to-tail. We don't know that yet. So I'm just going to show you 2 or 3. So as you can see, we did a bunch of mutations here. I'm just going to show you 3 interesting results here. So first is that the GRmon, so the one that has no known dimerization surfaces because we mutated it all, and it's mostly monomeric, at the array, the little cells that can form array, because this protein almost doesn't bind DNA, it is a dimer, okay? So once it binds DNA, it appears to, again, trigger this, what we call tetramerization. That suggests that the tetramer is a dimer of dimers, since in this case, all dimerization surfaces are gone and we still get, this oligomerization state. Another interesting thing is that we completely delete the ligand binding domain now. This protein is completely monomeric in the nucleus and it's a dimer at DNA, okay? So that suggests that the LBD is necessary for the formation of these higher oligomerization states. And even if we put the DNA mimic mutation inside this whole deletion mutant, what we get now is consistently with the idea that now the receptor believes is bound to DNA, the dimers, both in the nucleus and in the at the array. So to summarize, monomeric GR, we claim is not physiologically relevant. We believe the active GR, that means the DNA bound form, is likely a tetramer. And the dissociating model is completely outdated. And we need to stop trying to find better glucocorticoids by searching for monomeric GR. So with that, another summary will be a much, I guess, cleaner idea, where most of GR is dimeric, and upon DNA, it forms a tetramer. And DNA binding is essential for all activity of the receptor.